Welcome back students to chapter 9 Heredity and Evolution. In this video we are going to study about part 2 Mendel and his experiments. First let us study about Mendel. Gregor Johann Mendel was an Australian monk and botanist. He is regarded as father of genetics. He applied his knowledge of science and mathematics to his experiments on pea plant and established the principles of genetics. In 1857, Mendel began a series of experiments on pea plant to study the pattern of inheritance of various characters. He chose pea plant for three reasons. First, pea plants are self-pollinating. Second, they are easy to cultivate. Third, they have sharply defined characters. Mendel chose to study 7 different characters in pea plant. Each of these characters, such as height, seed shape, seed color, etc., had 2 sharply defined and contrasting traits. Like stem height had 2 traits, tall and dwarf, flower color, violet and white, flower position, axial and terminal, pod shape, inflated and constricted, pod color, green and yellow, seed shape, round and wrinkled, seed color yellow and green. He crossed a variety of pea plants carrying particular trait of a character with another variety having a contrasting trait of the same character. Mendel always used breeding plants. Pure breeding means homozygous for a trait. Plants can be made pure breeding by conducting controlled self-pollination for many generations. So let us discuss about Mendel's experiment. He selected tall plant and dwarf plant. They were taken as parents. He crossbred them, and in the first generation all the plants were tall. First generation plants were undergone self-pollination, and second generation offsprings were 75% tall and 25% dwarf. In the same manner, he conducted experiments in all other 7 characters. Terminologies used. Parental generation represented by P. They are the plants selected as parent. First filial generation or F1. First generation offsprings produced by crossing parental generation. Second filial generation or F2. They are produced by self-pollination of F1 offsprings. Gametes are represented by using circles and alleles are written inside the circle. So now we can see how we write the cross. So P represents parental generation. We write the phenotype and the genotype of the parental generation. Phenotype, tall and dwarf. So tall parent and dwarf parent are the selected plants. Genotype, he always took homozygous, dominant and homozygous recessive. Now we can write the gametes. Gametes are written by putting circles, and inside the circle we write the allele. So allele coming from tall parent is dominant allele, and allele coming from dwarf parent is recessive allele. Now when we cross the gametes, we get heterozygous tall plant. This heterozygous tall plant is the F1, first filial generation. When 2 traits of a character were crossed, the F1 plant showed only one of the trait. The other trait never appeared. The trait that did not appear in F1 reappeared in F2, but in 1/4 of the total number of plants. Mendel called the substance responsible for each trait as factors. Later factors were known as alleles. One allele came from one parent and the other from the other parent. When F1 were crossed, the F2 generation had 75% dominant trait and 25% recessive trait. The ratio obtained is 3:1 phenotypic and 1:2:1 genotypic ratio. Now let us study how we write F2 cross. F2 means F1 into F1. That means it is self-crossing. So the parents are heterozygous tall. Two types of gametes are produced because the parent is heterozygous. One gamete containing dominant allele and one gamete containing recessive allele. After that, we draw the Punnett square. On top of that, we'll write the gametes. One set of gametes showing male gamete and the other set showing female. So if you take gamete containing dominant allele, capital T, and cross it with another gamete containing dominant allele, the offspring will be homozygous tall. If you take a gamete with capital T and cross it with small t, the offspring will be heterozygous tall. Third offspring of the same, it is heterozygous tall. The fourth offspring, it is a cross between recessive T and recessive T. So, homozygous recessive dwarf character will be seen in the offspring. So, if you check the ratio, phenotypically 3 offsprings are tall and 1 offspring is dwarf. So, phenotypic ratio becomes 3:1. Genotype, 1 is homozygous tall, 2 are heterozygous tall, and 1 is recessive dwarf. So the ratio becomes 1:2:1. Monohybrid cross. A cross in which only one trait is involved is called monohybrid cross. Example, a cross between tall plant and dwarf plant, a cross between round seed plant and wrinkle seed plant, a cross between pink color flower plant and white flower plant. Now, laws of Mendelian inheritance. First law is law of dominance. In a heterozygous condition, always dominant allele is expressed in F1 generation. Allele which is suppressed is the recessive allele. So if you remember the cross, in the F1 generation we got tall plant. That means tall is the dominant character. This figure shows law of dominance. 3 genotypes are present, but phenotype is only 2. 3 offsprings are showing the dominant character and 1 offspring shows the recessive character. Second law of inheritance, law of segregation. It states that although the alleles of a trait remain together, they are separated during gamete formation. This figure illustrates law of segregation. So this is the parent, and in the parent the condition is heterozygous, but when the gametes are formed, both the alleles are separately moving into different gametes. They are not staying together. This is the meaning of law of segregation. Alleles separate during gamete formation. Dihybrid cross. A cross in which 2 different traits are used are called dihybrid cross. Example, tall plant, pink flowers crossed with dwarf plant, white flowers. Yellow and round seed plant crossed with green and wrinkled seed plant. Now, third law of inheritance, law of independent assortment. It states that each pair of alleles of 2 different genes is independent of the other pair in a dihybrid cross. So, let us study about dihybrid cross. First, we will take example, tall and round, dwarf and wrinkled. So the P is, that is a parental generation, is tall round crossed with dwarf wrinkled. We write the genotype, homozygous condition we will be taking. Then we write the gametes. In the gametes, both characters alleles are present. So one gamete is capital R, capital T and capital R. The other gamete is small t and small r. F1 produced is tall round in the heterozygous condition. F2 means F1 crossed with F1, that is self-pollination. So heterozygous tall and round crossed with heterozygous tall and round. We have total 4 set of gametes produced by a single parent. Total 8 gametes will be present. So if you see the gametes, one gamete will have capital T with capital R, another gamete will have capital T with small r, third gamete will have small t with capital R, and fourth gamete will have small t and small r. This is the 16 offspring that we obtain in a dihybrid cross in the F2. Offsprings obtained in the F2 of a dihybrid cross. Tall, round 9, tall, wrinkled 3, dwarf, round 3, dwarf, wrinkled 1. Tall and round, dwarf and wrinkled are parental characters. Two new varieties of characters are also found here, tall and wrinkled, dwarf and round. This ratio proves that law of independent assortment. The ratio of F2 is 9:3:3:1 for dihybrid cross. This is another example. You can go through this. Here I have taken the example of round and yellow, wrinkled and green. Same way I have made gametes F1 generation, F2, and then the gametes. Then we'll draw the Punnett square and find out the offspring. We'll get the ratio as 9:3:3:1, total of 16 offspring. Expression of traits. A gene contains information for making proteins in the cell. Proteins synthesized according to this information may be enzymes that catalyze biochemical reactions. Each trait is the outcome of several such biochemical reactions, each of which is controlled by a specific enzyme. If the enzyme is not produced because of the absence of genes, that particular reaction will not occur, and the trait resulting from this reaction will not appear phenotypically. Thus, each trait is controlled by a gene. So, DNA contains the gene. Gene codes for amino acid. Amino acid produce protein. Proteins will give rise to the enzymes, which is important for biochemical reactions, and because of this biochemical reaction we have a phenotype or a trait appearing. So if the DNA is not having the gene, no amino acid will be produced, no proteins will be there, which results in no enzymes, no reaction and no phenotype. Here we come to end of this video. Thank you for watching.